The overall goal of this procedure is to induce benign and invasive tumors in Drosophila, and then visualize them by dissecting the larval cephalic complex. This begins with setting up a cross between the desired strains and isolating the larval progeny bearing either the invasive or benign tumors. The next step is to dissect the selected larvae to expose the cephalic complex by first cutting them 2/3 of the way down, securing them, and then turning them inside out. Next, fat body and gut tissues are cleaned off, and then any nerves still attached to the ventral nerve cord and the larval epidermis are also broken. The final step is to image the cephalic complex or fix it for downstream applications. Ultimately, results can show GFP-tagged invasive or benign tumors through the use of fluorescence microscopy.
This method can help answer some of the key questions in cancer biology. For example, you may want to understand which genes could inhibit progression of tumors in an intact organism.
Once the required genetic stocks have been obtained, prepare a starter culture of the tester strain. Also prepare a starter culture for the tested stock. Cross 10 female virgins from the tester stock with 10 males from the tested stock. Allow the flies to mate and lay eggs for 24 to 48 hours at 25 degrees Celsius, and then transfer them to a fresh vial to produce more progeny. Monitor the cross vials.